Hello children, welcome to biology class. Today let's discuss the topic linkage and recombination from the lesson Principles of Inheritance and Variation. While discussing about meiosis in class 11, we had discussed about crossing over, isn't it? So, by recollecting the memories of that, we can discuss linkage and crossing over. Children, in dihybrid cross, when two genes were situated on same chromosome, the proportion of parental gene combination was much higher than the non-parental type. It was the observation by Thomas Hunt Morgan. Morgan attributed this due to physical association or linkage of the two genes and coined the linkage to describe the physical association of genes on the same chromosome. For this finding, he selected an organism, that is Drosophila melanogaster. And after his experiments, he found that the generation of non-parental gene combination during dihybrid cross is called recombination, and when genes are located on the same chromosome, they are tightly linked and show very low recombination. This is melanogaster, Drosophila melanogaster, male and female, fruit fly, that is a common name. And this fruit flies, Drosophila melanogaster, which were found very suitable for the studies conducted by Morgan. And what are the special attributes of Drosophila melanogaster? Children, you study the scientific name, okay? Drosophila melanogaster. And the special features are, they could be grown on simple synthetic medium in laboratory. Then the second thing, they complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. Then the third reason, there was a clear differentiation of the sexes. That means a male and female flies are easily distinguishable. From the diagram itself it is very clear, isn't it? Then the last thing, it has many types of hereditary variations that can be seen with low power microscopes. That's why he selected Drosophila melanogaster, the fruit flies, for his experiment. And Morgan attributed this that means his findings due to the physical association or linkage of the two genes, and coined the term linkage to describe this physical association of genes on a chromosome, and the term recombination to describe the generation of non-parental gene combination. Children, you can see this diagram in our textbook. This is that diagrammatic representation of cross A. Here you can see 98.7% parental type formed, and this is cross B. Here recombinant type only 1% of 3% were there. Here brown bodied red-eyed female, that is wild type. Here yellow bodied white-eyed male. So what this diagram represent? That means linkage means result of two dihybrid crosses. Cross A showing crossing between gene Y and W, whereas crossing between W and M. Crossing between W and M is shown here. Here. This is cross B. Then here dominant wild type alleles are represented with plus sign in the superscript. Children, please note the strength of the linkage between Y and W is higher than W and M, isn't it? Morgan and his group also found that, so even when genes were grouped on the same chromosome, some genes were very tightly linked. They showed very low recombination. See, this is cross A. Okay. What is the reason for this formation of 1.3% recombinant? Because some genes were very tightly linked. But that's at the same time you can see, and cross B you can see, yes, out of 100, 37.2% recombinant formed and 62.8% parental type 2 form. What is the reason? They loosely linked. They show higher recombination. Clear? Is it clear now? So, for example, he found that the genes white and yellow were very tightly linked and showed only 1.3% combination, while white and miniature wing showed 37.2% recombination tree. Here the genes are tightly linked, but here the genes are, yes, loosely linked. That is the reason. Okay. So now let's discuss the difference between crossing over and linkage. Crossing over, it leads to separation of linkage gene. It involves exchange of segments between non-sister chromatids of homologous chromosome. Here individual chromosome. Then 50%. The frequency of crossing over can never exceed 15%. But here more than a haploid chromosome number. It increases variability by forming new gene combination. Here it reduces variability. Now let's discuss the last topic of this lesson, genetic disorders. Children, we can divide genetic disorders into two categories as Mendelian disorders and chromosomal disorders. They are transmitted as affected individual is sterile. This is always dominant in nature. Okay. So what is the difference between Mendelian disorder and chromosomal disorder? Here in Mendelian disorder, it is due to alteration in single gene. At the same time here, these are caused due to absence or excess of one or more chromosome or abnormal arrangements of chromosomes. Clear? Here simply alteration happen. Here addition or deletion. Okay, or abnormal arrangement. And second difference, they are transmitted into generations through Mendelian principles of inheritance. But here, they may be recessive or dominant in nature. Examples, color blindness, then phenylketonuria. Example, Down syndrome, Turner syndrome. Now let's discuss three diseases. That means three Mendelian disorders. We will discuss hemophilia, sickle cell anemia. Hemophilia, then sickle cell anemia and phenylketonuria. Okay. First of all, we can discuss hemophilia. What is that? It is sex-linked recessive disease in which an infected individual, a minor cut lead to non-stop bleeding. Children, it is hemophilia is considered as a royal disease. This is because of Queen Victoria, who was a monarch of United Kingdom in 80s and 18 that century, that means 19th century, and was the carrier of the disorder, and she passed this condition to her son. Like that, several daughters were carriers and passed this gene to their royal families in Spain and Germany and Russia to their sons. Children, you just analyze this cross. See, this is a carrier woman. Carrier woman is represented by XH. Okay, and when she marries a normal man, and you just observe this Punnett square, XX normal girl produced, XY normal boy produced, XXH, that means carrier girl, and last one, yes, XHY, hemophilic boy. That means inheritance of hemophilia by 50% of the male children when the mother is carrier and the father is, yes, normal. Clear? Then second Mendelian inheritance, sickle cell anemia. Children, while explaining point mutation, we had discussed sickle cell anemia. Do you remember it? Okay. So, it is an autosome linked recessive trait in which mutant hemoglobin molecule undergo polymerization under low oxygen tension, causing change in shape of RBC. See? Change in shape of RBC from biconvex disc to elongated sickle-like structure. And what is the reason? This defect is caused by the substitution of glutamic acid by valine, and it is a single base pair change, isn't it? As a point mutation. So the substitution of amino acid in the globin protein results due to single base pair substitution of the 6th codon of beta globin gene from GAC to GUG, and it results the production of abnormal protein and the sickle-celled RBC formed. See, now you observe this cross. Here this is normal, carrier, and this is sickle cell anemia. Clear? Then third one, phenylketonuria. It's an inborn error of metabolism inherited as autosomal recessive trait. Here the affected individual lacks an enzyme that converts the amino acids phenylalanine to tyrosine. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and the other derivatives that results into mental retardation. Okay, so these are three: Mendelian inheritance. Now let's discuss chromosomal disorders. Failure of segregation of chromatids during cell division results in loss or gain of chromosomes called aneuploidy. The failure of cytokinesis leads to two sets of chromosomes called polyploidy. The first one, Down syndrome, due to the presence of additional copy of chromosome under chromosome number 21. Okay, so this affected individual is short-statured with small rounded head, furrowed tongue, partially open mouth, and mental development is retarded. Okay, so this is 21st trisomy, isn't it? Additional copy of the chromosome in 21. Whereas this is in Klinefelter syndrome, here it is due to the presence of an additional copy of X chromosome, that is instead of XY, XXY. Such a person have a overall masculine development, however feminine development. So now let's discuss the signs: a taller, less muscular body than males, and broader hips and longer legs, larger breasts, weaker bones, a lower energy level, smaller penis and testicles, delay in puberty, then less facial or body hairs. Then last one, Turner syndrome, caused due to the absence of one of the X chromosome. That means 45th with X0. See, here one chromosome is missing. So for X0 condition, such females are sterile as ovaries are rudimentary. They lack secondary sexual characteristics: small mandible, high arched palate, low set ears, low hairline, bicuspid aortic valve. Then lots of characteristics are there: ovarian dysgenesis, etc. Okay, so that is Turner syndrome. 45, that is X0. So Klinefelter syndrome, then Turner syndrome, and Down syndrome. Down syndrome is 21st trisomy. Clear, children? That is about chromosomal disorders. Okay, so that is about the lesson Principles of Inheritance and Variation. Okay, you read the textbook thoroughly. Okay, children, tomorrow let's start a new lesson. That's the last lesson of our biology textbook. Yes, molecular basis of inheritance. Okay, children, study well.